connected organelles. And when I say connected, they could be connected physically, or they could just share parts of that membrane system. So this is a list of everything that's in the endomembrane system. Of course, that includes your plasma membrane, but it also includes the nuclear envelope. Remember last class we said the nuclear envelope was actually made of membrane. The endoplasmic reticulum, we call that the ER, rough ER, smooth ER. The Golgi apparatus. And then we also have what are called lysosomes and vesicles. And all of these are part of that endomembrane system. And as a whole, it regulates protein movement within the cell, as well as what is removed or pushed out of the cell. Okay? And so, again, all of these structures are made of plasma, of the membrane, of that phospholipid bilayer. But remember we talked about last time that the phospholipids in that bilayer are not physically connected. They're not covalently bonded to each other. The only thing that's keeping those phospholipids in line with the other phospholipids is the very simple hydrophobic interactions of the lipid tails. Right? The lipid tails hate water, and so because of that, it all sticks together. But it's not bonded to itself. Right? And so what that means is that these phospholipids can move around between all of these different structures. And so all of them are related, and we call that the endomembrane system. So the membranes can be related either directly by physical continuity, or they can move sections of membrane between or among themselves. The membranes in each of those different structures are not identical. They are individualized based on the membrane's function and the needs of the cell at that particular time. And so the membrane itself changes composition as it moves from location to location between these different structures. And so it can change in thickness. When I say molecular composition, the kinds of proteins that are in those membranes, how much cholesterol is present in those membranes, the types of chemical reactions that they facilitate, none of those things are fixed. And so as these membranes move from the nuclear membrane to the Golgi, to the ER, back to the nucleus, all of the membrane structures can adapt to whatever the needs are in that spot. And that's what this is saying here. Those membranes can be modified at several times during the life of those phospholipids. We'll start by looking at the endoplasmic reticulum. The endoplasmic reticulum, or the ER, has two different forms. We have the smooth ER and we have the rough ER. And the difference between the two is the presence of ribosomes. And remember that ribosomes are the sites of protein synthesis. They're making the proteins, they're taking the mRNA that was produced in the nucleus, and they're actually producing the proteins that that mRNA codes for. And so when we look at our endoplasmic reticulum, we see that it is physically continuous with our nuclear envelope. So here's your nucleus with those pores, physically continuous to our endoplasmic reticulum. These dots are your ribosomes where we could produce proteins. And so we're going to look first at our smooth ER, which is this portion over here, no ribosomes, and then we'll look at our rough ER, which is the part that has ribosomes. Smooth ER does a couple of different things. Smooth ER, for the most part, synthesizes lipids. So remember that you produce lipids, right? You can produce fats. That occurs in your smooth ER. But the other thing that the smooth ER can do is metabolize carbohydrates and detoxify drugs and poisons. How does it do that? Well, a lot of times, in order to flush toxins from your system, you may have heard that phrase before, flushing toxins from your system, we really just have to make them more soluble because you are made of water. And so as long as we can make these things more soluble, the water that you drink will remove them from your body, but they have to be more soluble. So how do we make them more soluble? We add a hydroxyl group, right? Remember hydroxyl? Hydroxyl is going to have an oxygen at the end. Polar, nonpolar, polar, oxygen. That means water is going to be able to dissolve it easier. So we add a hydroxyl to the end, makes it more soluble, and it flushes from the body. But here's the trick. It's the smooth ER that does that. And so repeated use of drugs causes that smooth ER to proliferate and increases the rate of detoxification. So this is when you may have heard of people who are possibly alcoholics or drug addicts and things like that, where it's like they have to consume so much more of that substance in order to get high or to get drunk. It's because their smooth ER has proliferated and their cells are so good at detoxification that they're detoxing the substance before they get high or before they get drunk. And so that's why so much more is necessary to get that same high. And that's the problem with a lot of drugs and alcohol that we see in society, is that the people who use them regularly The smooth ER also stores calcium ions. This matters a lot with neurons, and we'll get to that in Unit 7. That rough ER has fewer functions, but is certainly no less important. The rough ER is the site of protein synthesis where those proteins are going to most likely be exceeded or kicked out of the cell. So stuff that's going to be excluded, right? We're going to push it out. And so the ribosomes that are attached to the rough ER grow polypeptide chains directly into this interior portion of the ER. Why does that matter? Well, remember we talked about proteins and we said proteins have an ideal temperature, pH, hydrogen ion concentration, right? All of those things have to be ideal for a polypeptide to take the appropriate functional shape of a protein. So in these little, they look like pita breads, but in these little pita breads, the lumen portion here, the cell can create an ideal environment specifically for the protein's shape. So we can say, well, I need a protein that's going to function at a pH of 6. Out here is a pH of 7. So in here, we're going to create a pH of 6 and have a perfect little microenvironment for whatever protein it is we need to build. And so it's stuck in here, we create that little microenvironment, and now that polypeptide is able to fold properly into the protein that it needs to be as it's moved around the cell. Okay. So that's one of the functions, and then it's also really good because what happens is, once those proteins are produced in here, each of these little guys, remember the membrane system, we can pinch off a little piece of our Golgi. Once we do that, now it's called a transport vesicle. Remember the vesicle on top of the guy's head who's walking around in the cell? Remember that? Okay, so now we're going to take this transport vesicle and we're going to take it to our Golgi apparatus. Okay, yes?
So the transport vesicle could have a different pH than
the inside of that transport vesicle? Yes, it can. Absolutely can. So it's keeping these proteins separate, okay, over here, from the ones that are being produced by the free ribosomes that are floating around in the cytosol. So we have some ribosomes that are actually attached to the ER and other ribosomes that are free-floating in the cell. The ones that are free-floating in the cell tend to produce proteins that are used within the cell. The ones that are attached to our ER here is producing the proteins that are going to be excreted from the cell. Okay. Or that are going to stay locked in the membrane. Either of them. So here's that vesicle coming in to visit our Golgi. When you were taking classes in CP and honors, you probably called this the post office, the shipping and receiving center. Okay, now we're going to call it the Golgi apparatus. The CIS, the cis phase here, is the receiving end. The trans phase here is where they depart. Two big things happen in the Golgi apparatus. One, protein processing can occur. If we're going to have a protein that requires quaternary structure, this is where it's all going to come together. Sometimes we can modify tertiary structure, but for the most part, that was taken care of in the endoplasmic reticulum. The other thing that's happening is that the Golgi apparatus is identifying that protein, figuring out where in the cell it needs to go, or if it needs to be excreted altogether, and then essentially giving it a molecular ID card and saying, hey, you need to go to that membrane, or you need to go to that organelle, or you need to go to that structure. And it literally gives it a molecular ID tag that sticks out of the outside of these vesicles. And then this vesicle is going to go to that motor protein, the guy who's carrying it around with giant shoes. It's going to go to that motor protein, and the motor protein is going to be like, okay, I got this molecule, this is my zip code, it's like a zip code, and now I'm going to figure out where in the cell does it need to go. Okay. So what the Golgi is doing over here is it's saying, okay, these are the proteins I've got, are we good to go, or do I need to modify it, and now where in the cell does it need to belong? And out it goes. Okay? Yes? Are you scratching your head?
Oh, yeah.
That's great. Okay. So we also have something called lysosomes that are a part of the endomembrane system. Lysosomes are only found in animal cells, and they're used to digest macromolecules. Remember, that's achieved by hydrolysis. So we're adding that water molecule and breaking apart that polymer. And so here's the trick. The whole cell is made up of these polymers. And so if these enzymes that hydrolyze polymers are floating around in the cell, the whole cell is going to fall apart, right? Because these are the enzymes that break apart macromolecules. We need the macromolecules to stay intact unless we've identified them as broken or non-functional. And so what happens is lysosomes contain the enzymes that take macromolecules and break them down. Here's what is so awesome. And so this is to Caleb's question a moment ago. He said, so the inside of that vesicle could have a pH that's different than the cell. And that's exactly what's true in lysosomes. Lysosomes have have an optimal pH that, or the interior portion of the lysosome has a pH that is lower than the outside cell. And the enzymes that engage in this hydrolysis have an optimal pH that is also lower. So the optimal pH is about 5 for these proteins. You don't need to know that. But it's about 5 in most organisms. And so the inside of that lysosome has a pH of 5. The ideal pH for that enzyme is 5. But if that lysosome for some reason were to break apart and that protein were to get out of the lysosome, what would happen to it? It would denature, right? And if it denatures, is it going to function? If it denatures, it's not going to function. That's good news. Because we don't want escapee enzymes running around the cell taking apart stuff that shouldn't be taken apart, right? And so we have lysosomes that have a much lower pH because we're going to have that protein that is optimal at a lower pH so that if it gets someplace it's not supposed to be, it doesn't just dismantle the cell. That's good. I think that would be terrible. So here's what that looks like. So first of all, there's 2 reasons why we might engage in this with our lysosome. Okay. So here's your lysosome, digestive enzymes, a lower pH on the inside. Phagocytosis we pulled in, so this is called cellular eating. We'll get to that in another chapter. Okay, so we've taken in something from the outside. The lysosome binds with it here. This is all membrane. See, this is an infolding of membrane. This is membrane. Those phospholipids join together. And now these enzymes come here and break it apart. Now we can use that. They're just macromolecules. Now there's monomers. Hijack from something else. Okay, now we're going to build proteins or carbs or whatever we want to do with them. Okay, so we can digest them. So we also might use it. This is called autophagy, self-eating. The lysosome, sometimes cells can be old, right? I mean, you are the cells that you were born with, and they will be the cells that you die with because they just keep dividing and dividing and dividing and dividing. So the mitochondria, at some point, they wear out, okay? And as is true with all of your cellular structures, they could get, damaged or whatever. The lysosome would attach to it and break it apart. We'll get to peroxisomes a little bit later on. Break it apart. And now we again have monomers that we can use in order to co-opt those for another purpose. Okay, vacuoles. Sometimes vesicles get gigantic and we call those things vacuoles. They're derived from the ER and the Golgi, membrane bound, okay, part of that endomembrane system, and they're surrounded by a selective membrane. But the solution that's inside the vacuole is different from the cytosol. Okay, it's different from the cytosol. So you could have food vacuoles that are formed by phagocytosis, where the food just is going to hang out there until the cell actually needs it. We could have contractile vacuoles. Think about single-celled organisms that live in fresh water. Fresh water is constantly going to be going into those cells, and if it's an animal, it's going to explode, which would be really unfortunate. And so if you don't want to explode and you're a single-celled animal and you're living in fresh water, what do? You pump that water out as quickly as you can, and you use a contractile vacuole. Contract means to squish, right? It's a vacuole, it fills up with water, and then squishes, and it all leaves. Okay? I can't say how much like SpongeBob it is, I'm not familiar, but I'll take your word for it. It's like SpongeBob. Got it, noted. And then we also have central vacuoles in plants, and this is what it looks like, and you guys will see that when you look at Elodie next time I see you. You'll actually see an area of the cell that looks empty. Here are all of your organelles, okay? You have a region that's like, oh, that's weird. Well, here's the deal. It's a vacuole that's surrounded by a membrane. The cell can put stuff inside, usually in plants, inorganic ions, but here's the thing. Vacuoles don't count against your volume. Remember we talked about surface area to volume ratio? And the problem was that the volume grew so much faster than the surface area? Well, plants can just store extra stuff in here, and because it's metabolically inert, it's not metabolizing, this doesn't count against your volume. Because the reason the surface area to volume ratio mattered was because we had to have enough surface area to absorb the reactants and get rid of the products of all of our chemistry. Well, if that's not happening in here, it doesn't count against you. Okay, so our central vacuole can be used for that as well. So here's the diagram. Okay, puts the whole thing together. I'm not going to talk you through it, but I put it in here because I want you to understand that when we talk about the endomembrane system, we're talking about all of these different structures. It's in your notes questions. The this particular diagram, and I want you to be able to practice writing out the process of what happens beginning in the nucleus all the way through. And you can just pick a protein that you're going to synthesize, be like the directions for the protein synthesis begins in the nucleus in the form of a chromosome, and then the mRNA leaves, right? And that's all the whole process. And how does the endoplasmic reticulum get involved? How does the Golgi apparatus get involved? What's going on with the vesicles? Okay, the whole thing. Try to put the whole thing together. There's a whole page on this diagram in your textbook that leads you through the whole thing. Okay, so it might be a good idea, especially if you haven't taken bio before, to read the diagram, make sure it makes sense to you, put it away for the day, come back, be like, can I reproduce this? Can I see how all these different parts fit together? I don't know. All right, 4.5, moving on. So now we're going to talk about mitochondria. Yeah, there it is, right? You found it. Mitochondria and chloroplasts. Let's talk mitochondria and chloroplasts. So you're not going to call the mitochondria the powerhouse of the cell. You're going to call it the site of ATP synthesis. Okay?
Which is the powerhouse.
Okay. What happens in your brain is one thing. Once you write it in paper, it's going to be something different. All right. So all organisms are energy transformers. Okay. I eat energy in the form of food, and I transform that food energy into ATP, and that ATP is then transformed into energy for me to move around, kinetic energy, right? As I go ahead and I do my stuff, thermal energy, I lose a lot of energy as heat. Okay. Plants get energy from the sun, turned into sugar, and then use the mitochondria to take that sugar and also turn it into ATP. Okay. So we're going to be looking at these two structures here. They are not a part of the endomembrane system, even though they are surrounded by membranes. The reason they're not considered to be a part of the endomembrane system is because their membranes are not continuous with the structures we looked at earlier. So remember that defining characteristic was the ability of the phospholipid bilayer to literally move from the Golgi to the ER to the nuclear lamina to the plasma membrane, and to make all those moves. These two structures, the mitochondria and the chloroplast, do not share their membranes with other parts of the cell. So they are not a part of that system, even though they are membrane bound. First, the mitochondria, the site of ATP synthesis. The outer portions of the mitochondria are smooth, and the inner are convoluted with a lot of folds. Okay. Why does that matter? Loads of surface area. The business part of the mitochondria, the production of ATP, happens across a membrane, and it happens across this inner membrane. And so in order to be as efficient as possible, the more membrane we have on the inside means the more ATP can be produced. That's why we have so much inner membrane in the mitochondria. We call this place the matrix. These infoldings, the cristae, and then this space in between them is the intermembrane space. Within the mitochondria, we have free ribosomes. Mitochondria has its own DNA, and it is circular. So that will come up again later on in the course. Similarly, chloroplasts, which are converting solar energy into glucose, along with the building block carbon dioxide. Chloroplasts have very similar structure in that we have a double membrane with an intermembrane space. We have our circular DNA, and it has its own ribosome, so it produces its own proteins. Instead of having our loads of membrane being our infoldings here, instead we have extra membrane in the form of thylakoids. They look like hamburger buns. When you stack the whole thing together, we call it a granum. The fluid on the inside is called the stroma. So that's the structure of your mitochondria.
What's
The fluid. The fluid that's in here. Okay. Peroxisomes, let's also get to peroxisomes. Peroxisomes are kind of weird. They're not chloroplasts and they're not mitochondria. They're something different altogether. But they're these very specialized metabolic compartments. They only have a single membrane. They're typically affixed to, here's a chloroplast, here's a mitochondria. They tend to be affixed to one of them, though not always, which is why it's in this unit. But what they do is contain enzymes that remove hydrogens from molecules and transfer them to oxygen. And that forms H2O2, which is hydrogen peroxide, which is why we call it a peroxisome. And this is detoxification. Okay, for example, alcohol is detoxified by removing a hydrogen and producing hydrogen peroxide. The trick is hydrogen peroxide will kill you. It's very toxic. And so once a peroxisome produces H2O2, then what it's going to do is break this down into water. And so in this way, the cell isn't actually damaged by the H2O2 that's produced. It's producing the H2O2 in order to detox stuff, and then it's dealing with the hydrogen peroxide so that you don't get hurt by it. I bring this up because in Unit 3 we'll do a lab that deals with peroxisomes. Next we're going to talk cytoskeleton. The cytoskeleton is pretty much ignored when people learn about the cells in lower level classes. In essence, the cell itself has shape. And so it's not just going to be like water that splatters on the table and takes whatever shape of its surroundings. It does have its own shape. And it's very similar to if you ever go camping, the tent that you would put up. You have the poles that literally hold the whole thing up. But then do you remember when we were watching the video and there was the dude who had the thing, right? He was transport protein, right? He had his vesicle on his head. He was walking on a road. Do you remember that in the video? Yeah. Those roads are these green things, the microtubules. Okay, they're actually stiff. They're actually holding the cell apart. And then the nucleus is also super important, right? That's where all your DNA is. And the nucleus is going to have to stay in its proper shape as well. And so we're going to actually use what we call intermediate filaments for that. So we have 3 different types of polymers, really, that are going to help keep our cell shape. We have the microtubules, which are the green ones that you see here, the microfilaments, which are the red, and the blue are going to be our intermediate filaments. We're just going to look at these briefly. No matter what, when we talk about the cytoskeleton, no matter which structure we're looking at, they all do mechanical support and they all maintain shape. So we're just going to keep that in mind. If you're ever asked, provide one function of microfilaments, microtubules, intermediate filaments, I don't care, maintain cell shape, done. Always going to be correct. Okay. And again, it works like a dome tent. They provide balance by those opposing forces. And they provide anchorage for a lot of organelles. Okay, because we don't want the organelles moving around, we need to have them distributed through the cytosol. Cell motility, right? This is what we saw in the video. We have a motor protein that uses ATP to move vesicles around, and they use it kind of like roads or a highway. I think the cytoskeleton is used for that as well. Those 3 main types of fibers, like I just showed you in the picture, microtubules, microfilaments, and intermediate filaments. Microtubules are tubes, and they are hollow. Okay? These are the thickest ones. See how there's space in between them? Microtubules. They're made up of what are called tubulin dimers. So this part is the dimer. We have an alpha and a beta subunit. This is the dimer. And as we add them in this spiral fashion, okay, it just gets longer and longer and longer. And then we can take them apart. And you saw that in the video when I was like, look, polymer! Okay, that's what we were building. We were building microtubule. Okay, so that's how they're able to change the shape. What are our functions of our microtubules? Maintain cell shape, always. It's always one. Cell motility, we may have heard of cilia and flagella. Cilia and flagella are made of these microtubules. Chromosome movements and cell division is another one. We'll get to that a little bit later on in this unit. And then organelle movement. Microtubules are what we use to build centrioles. 2 centrioles make up a centrosome. You may have remembered this being called the microtubule organizing center when you learned about mitosis back in CPR on our bio. But when when we're going through the stages of mitosis, microtubules shoot out of the centrioles to attach to the chromosomes and move them around. We'll get to that later on in this semester. Microtubules in special arrangements can do things like cell movement. Flagella or cilia are good examples of this. Flagella, there's usually 1 or just a few flagella per cell, usually used for movement, and it like flaps around like a whip. Okay. Cilia, there tend to be many, many, many cilia, although not necessarily all the way around the cell. This is a paramecium, pond water, they live in pond water, and the cilia are used for movement, so they just beat. You are full of cilia in your trachea. Here, only this surface of the cells have cilia, but that's what you have there. And the reason you're lined with cilia here is they're constantly beating. This is a dust particle that you've inhaled. And so the job of your cilia is to stick to these things and then kick it out. Okay, we gotta get rid of it.
How does it get kicked out?
So it just gets floated up, and then you cough, and then you swallow it and digest it. Delicious. We have microfilaments. Microfilaments are thin, solid rods. Thin, solid rods built of a protein called actin. These are really, really, really strong. Again, maintain cell shape. They do this by bearing tension, so this is what we have on the outside of that cell. Also involved in cell division of animal cells, so again, cell shape is from the outside. Microfilaments are what actually create that cleavage furrow, if you remember that last step when we pinch the thing in half. But also used for cell motility, muscle contractions, amoeboid movements, cytoplasmic streaming in plants, okay, that kind of stuff. Microfilaments. Intermediate filaments are named for their diameter, they're in the middle. These are really, really, really probably our strongest. And these are the ones that anchor the nucleus where it needs to be and give the nucleus support. It forms that cage around the nucleus, right, the nuclear lamina. Those are your intermediate ones. Just a couple of more slides here. We're going to talk about extracellular components and connections between cells. Coordinate cellular activity. So the cell wall in plants is actually an extracellular structure. The boundary, the exterior boundary of the cell is the cell membrane. The cell wall is outside the cell membrane. Okay. So the cell wall gives protection to plant cells, also present in fungi and many types of prokaryotes. The cell wall maintains shape and prevents excessive water uptake. We'll deal with this later on in this unit. What does that actually mean? On a whole plant level, the cell wall is going to actually hold the plant up against the force of gravity. So if you look at our classroom plants, they don't have a skeleton. They don't have bones. What allows them to not be limp like cooked broccoli is cell walls. Okay? Cell walls and turgor pressure. So even though animal cells don't have cell walls, they do have a pretty elaborate matrix called the extracellular matrix outside that cell. Can be attached to integrins in the plasma membrane. Okay, here they are. Integrins are these proteins. And this whole thing out here is extracellular matrix, an extracellular fluid that literally holds it and pulls the cell out. Neighboring cells can actually communicate with each other, but they're joined in a couple of different ways. They can have physical contact, but then we can also have something in plant cells called plasmodesmata that are literally little roads that allow the fluid here and the dissolved ions here to just go next door. Go here, go here. Now we're not going to move major organelles. The nucleus isn't just going to get lost. Okay. We're just moving what's in that cytosol. Okay. We call those plasmodesmata. What does that achieve? Essentially unifies most of a plant into one living continuum. Okay, so each cell operates on its own, but all of those cells are communicating with one another. Animals have three types as well. We have tight junctions, what are called desmosomes, and gap junctions. Gap junctions are most similar to the plasmodesmata. They're just the animal cell version. And literally, again, just let ions, small molecules flow through from space to space. Desmosomes and tight junctions are a little bit different. You can imagine desmosomes like a stapler. Two cells that are right next to each other, okay, can literally staple together in locations just to make sure that they're not relocating. But then we also have tight junctions. Imagine tight junction like double-sided tape. So it goes between the cells all the way around, and it's sealed anywhere we have these tight junctions. This will often create waterproofing in cells. That's what allows your skin cells to be waterproof. Okay. Are those tight junctions? With only 9 minutes left in class, so I did load the tour of an animal cell and the tour of the plant cell. These are the BioFlix tutorials. We're not going to take time to watch them right now because I want to do 3 multiple choice questions after test. But these are linked to your cell mastery biology assignment. So you can watch them there. I don't know if when you guys download my PowerPoints, if you can actually watch the videos or if you have to, I don't know if they work or they don't. But they're linked there so that they're there. Yes?
Like all of like the pictures and like diagrams on mastering biology, like it says you need a plugin, but it doesn't tell you what plugin.
So you have to go, so somebody came to my office and we did it together. You have to go and it'll tell you, because there's so many different browsers,
Yeah.
it'll tell you exactly how to check and what you need for the different browsers that you have.